Well, Monsanto's high lysine GM maize has bit the dust. Basically, yep, great, yeah? And you know what else? They've withdrawn it from application from the EU in secret some 6 months ago. And the EFSA, the European Food Safety Authority, and higher authorities as well, possibly connived in keeping it a secret. The reason they gave was that it was for commercial reasons because they don't really think there's a market. But actually, for once, EFSA was doing its proper job. They said there were certain safety issues and they wanted more data. And very likely, Monsanto was unable to supply that. Anyway, so what I want to tell you is that there are general hazards of GMOs, which are general to all GMOs because of the way that they are made. And then there are specific hazards to each GMO because basically of what's in it and because of all the hit and miss business involved in making them. So the best definition of GMO that I can think of is an organism with synthetic genetic material inserted into its genome. Its genome, the genome is just the totality of its genetic material. And it's made in the laboratory with sterile techniques, which also means no sex involved, okay? And here is a quick lesson in biology. All organisms, such as a maize plant, is made up of tissues and cells, and as you go down the scale with an increasingly powerful microscope, you will see that the cells have a nucleus in the middle, and this nucleus contains chromosomes. The chromosomes are little structures, and the totality of the chromosomes is your genome, because each chromosome, if you unwind it, is a long, very, very long, thin thread called chromatin. And if you strip the chromatin of proteins, you end up with DNA, which is the genetic material. And that's what gets chopped and changed in genetic engineering. And the rationale behind genetic engineering is basically what is called the central dogma of molecular biology, due to the Pope of molecular biology, Francis Crick, who shared the Nobel Prize with Jim Watson for working out the double helical structure of DNA. So according to the central dogma, the DNA carries all the genetic instructions for making an organism, and individual messages in the DNA is faithfully copied into RNA, which is another kind of nucleic acid, and then the RNA is translated into protein according to the so-called genetic code, and the protein determines a particular trait of the organism, as for example, herbicide tolerance or insect resistance. So that's basically what's inspired genetic engineering. And if things were as simple as that, a kind of a linear causal chain with one gene determining one character, then genetic engineering, genetic modification would work perfectly every time. Unfortunately, things are not that simple, and that's where the problems lie. So these are the general hazards of GMOs. And there are three general kinds. The first is due to uncontrollable, unpredictable effects, because the genetic modification process is a hit or miss business. So what it can do is that not only do the GM inserts, the synthetic DNA that you put in, it can get rearranged, truncated, or duplicated. It can also scramble the host genome rather badly. It causes widespread mutations or changes in the DNA. They can inactivate genes or they can activate genes, including those associated with cancer. It can create new transcripts, new RNAs with regulatory roles, creating also, it can create new unintended proteins that could be immunogenic. It could create new metabolites that may be toxic. And it could activate resident viruses. Remember the lecture you just heard before lunch? They're there all the time. They create new viruses also by recombination of the viral genes in the GM insert with those in the host genome. The second kind of hazards are due to potential effects of the transgenic proteins introduced. So these proteins, even if they were harmless in the original organism, could become immunogenic, could become allergens or bad immunogens. And they could create new proteins that may be immunogenic as well. The third kind of hazards are effects due to the GM insert and its instability. Basically, you know, the synthetic genes, the synthetic genetic material you put in is not stable at all. They could rearrange as you grow the GM crop in subsequent generations. So all this risk assessment, even if they do it properly, will be useless anyways. And because there will be further unpredictable effects generated, and most of all, and most underplayed, horizontal gene transfer. Basically, the synthetic DNA, being unstable, could jump out again and jump into the genome of organisms that interact with the GMO, including humans eating the genetic material, for example. And this horizontal gene transfer and recombination is a main route to spreading antibiotic resistance genes and creating new viruses and bacteria that cause diseases. It could also create mutations in the genomes of cells to which the GM inserts are transferred, including cancer. Now, just to take the example of the high lysine maize, what are the serious hazards that are unique to it? Well, this high lysine maize has 50 times more free lysine. Lysine is an amino acid. And 110 times more lysine derivatives than normal. Now, this is basically because normally in a balanced, normal organism, they don't accumulate these free lysine or derivatives because they are not very good for you. Lysine and its derivatives combined with sugars, also present in high concentrations in maize, to form so-called advanced glycoxidation end products, AGEs for short. And AGEs are bad news because they are implicated in many important diseases, including Alzheimer's, diabetes, and related autoimmunity, cancer, heart disease, kidney disease. So, now, these are not just theoretical risks. There's now a stack of evidence built up on hazards of GM food and feed or other exposures to GMOs. And they come from a combination of scientific experiments carried out in the lab and also from farmer witnesses in the fields, which our regulatory system, our regulator says, well, we can't take that into account because they're not in peer-reviewed, published scientific literature, which is basically, you know, just an excuse for doing nothing. I've color-coded it. The pale blue ones are the lab experiments, and the pale blue ones with asterisk are actually lab experiments done by the companies, but the data reanalyzed by independent scientists. Because if you leave it up to the companies, they will say, oh, well, okay, this is statistically significant, but it's biologically not meaningful, and we can just ignore it. Therefore, our regulators then accept that as safe. That's what EFSA has been doing quite a lot, and our food safety, our FSA, as well in the UK. And the white ones are all farmer witnesses. It's wrong to call them anecdotal evidence because these are real witnesses. In the fields, farmers in India finding their sheep, cows, and goats dead and very ill from eating the crop residues of the BT cotton, the GM cotton. And unfortunately, because most of these farmers are homeless, they don't actually have, they're completely the kind of the underclass, and nobody takes any notice. But so they have been ignoring all these deaths. And in the Philippines, again, that had been happening, and also in Germany, Hesse, Germany, a farmer actually fed BT maize to his cattle because he really prided himself on being very clever, and therefore he wanted the best for his herd, and he really got into trouble. A dozen cows died, and others in the herd had to be sacrificed. And, all these evidence have been published in our magazine, and I will just show you a couple of pictures containing, well, these pictures are actually given to me by the scientists themselves. This one, this is a lovely picture of Dr. Irina Ermakova. She is a senior scientist of the Russian Academy of Sciences, and she has done experiments feeding GM soya to rats and then letting them reproduce. And she found excessive deaths, severe stunting, and those offspring that survive into adulthood were sterile. I mean, you don't actually have to do the statistics. You can just look at the difference between the litter. You can guess which one was the litter that was fed GM soya. Okay, this is famous Akpat Pustai, Dr. Akpat Pustai and Dr. Stanley Ewing. Yes, I think they deserve applause for what they've done for us. They managed only to publish their results partially in The Lancet. The Lancet editor was intimidated, but he published it anyways. But they couldn't publish their whole results. You know why? Because their results were impounded by the Rawood Institute where Pustai worked. And what they found was that when they fed GM potato to young rats for just 110 days, they found that it damaged every organ system of the rats. And the stomach lining grew twice as thick. So, there you are. And there have been new evidence found since then, but I'll just show you a summary slide. I mean, look, this is really just very bad because there is basically these scientists who dared to do research are putting themselves at tremendous risks and sacrifices because uniformly they have had their grants cut, they lose their jobs, or they get intimidated as well. And the Monsanto hotels of the world are out to not only control the flow of scientific information, they want to control the kind of science that could be done at all. And which is why we exist. The Institute of Science in Society, we want to reclaim science for public good, and that's a very important project. Now, because, the combination of the brave new world of bad science and big business It's literally lethal, okay? It not only hurts us, not only hurts food safety and so on, it's killing farmers in India. And the rural affairs editor of the Hindu, the Indian newspaper, he calls it the largest wave of suicides in history. Between 1997 and 2007, nearly 200,000 farmers committed suicide. And most of it, the larger part of it, was due to GM crops, GM BT cotton, introduced since 2002. So, now, you know, those people who are responsible for promoting GM, who are foisting GM crops on the third world, basically, they are committing a heinous crime against humanity. And I'd like to think that there is nothing more sinister than that they have been adhering to an old, obsolete paradigm. And I'd love to give them this lesson in genetics, in the new genetics, leading up to the science of the organism, so that they can see and mend the errors of their way. So, here it is. Why is GM, genetic modification, both useless and dangerous? Basically, because it's based on the wrong science. Since the 1980s, genetic engineers in doing laboratory experiments have already discovered what is now called the fluid genome. There is no linear, one-directional causation, according to the central dogma. The geneticists have been turning genetics upside down in the laboratory since the 1980s, and they discovered that there is a circular causation of basically the genome and the organism as a whole is engaged in constant conversation with the environment. Everything is interconnected. The genes, the gene products, everything. And these constant feedback with the environment not only tells genes where, when, and by how much they should turn on and turn off, they also change the genome and the genes themselves, okay? And so, you know, the RNA, instead of just like, you know, transcribing faithfully the genetic message, they are like undercover subversive agents. They choose what to copy, and then they change the message in many, many different ways, or they can destroy it before they transmit it, or else they can even rewrite this holy text, you know, the DNA. You can see the analogies between a fundamentalist religion and the central dogma of molecular biology. So that is just completely wrong. Now, this has real practical consequences, and they are very important consequences. Basically, there is now a new discipline of epigenetics. Basically, because there are so many mechanisms that have been discovered that mark and change the genes according to an individual's experience during critical periods of their development. And these influences are actually passed on to future generations. So, so-called Lamarckian inheritance of acquired characters is rife in the molecular world. And not so long ago, that was the biggest biological heresy you can commit, and you can get excommunicated. You know, it's very, very serious. Anyway, here again, if you get exposed to environmental toxins, guess what? Again, during critical periods, not only you get predisposed to cancer, but your children and your children's children may also get the same thing. And that's called epigenetic toxicology. These are very well documented, especially with animal experiments. 4 generations after exposure, the effect still lasts. Now, you know, they told us lies. They said, if we sequence the human genome, we can tell you how to make a human being. Embarrassingly, they couldn't even, decades of sequencing the human and other genomes, and there's embarrassingly little to show for it. They haven't even been able to identify all the genes that predispose, or any genes, basically, that predispose us to the most serious chronic diseases. And let alone, you know, genes that give us high IQs or other undesirable characteristics. And if you hear anything like that, you should say, rubbish, that's what it is. Anyway, so what happens? All the evidence, the overwhelming evidence indicates that it is the environment that and epigenetic modifications in the environment that overwhelmingly determine our health and well-being. Not only that, we now know that early development and maternal care, early nutrition are very important for determining our physical and mental health. And therefore, it is of utmost importance that in delivering primary health care, we should take these into account. And I won't have time to go into this, but basically I agree with the speaker just before lunch. We need a new kind of medicine, and we're tackling that too in ISIS. But let me just go back to the new genetics of the fluid genome to emphasize why it is that genetic engineering is so dangerous. Basically, you know, the molecules, the genes, gene products and everything are engaged in a molecular dance of life that has evolved to perfection over billions of years of evolution. And if you put in these synthetic rogue genes, they don't know how to behave. They don't know the language, they don't know the score. I call this molecular dance of life quantum jazz, by the way. I'll explain later why. And these synthetic genes, furthermore, have a tendency to run amok. Basically, they would disturb everything, they jump in, jump out, unscramble the genome. And that's why it is so dangerous. So, the jury is out. We have no time to waste. We should ban GM crops now, especially, we don't need to ban it in the laboratory, but definitely we do not want it released into the environment, into our food chain. We've summarized all this evidence: less yield and income for farmers, bad for the biodiversity, more pesticides and water use, more dependency for farmers, more suicides, more vulnerable to pests, diseases, and climate extremes. And there are these outstanding safety concerns. Good. End of GMOs. Okay, now we go on to the real business of this paradigm change, paradigm shift. To understand the scientific revolution that's going on at the moment, which is very important, that can actually take us out of the multiple crises, we have to go back to the European Enlightenment, which gave us a lot of good things. Basically, it was the age of reason. It says, you know, and it also transferred the power of creation from God to nature, which we can now understand by scientific inquiry. But it also gave us a view, a predominant view of the world as machine. And this is the dominant paradigm, okay? I call it DOMO for short, because it actually is in everything, not just biology. It's in our economy, it's in our political, social, and educational institutions. It runs our industry, everything. We just have these assumptions. Our language is like that. It's kind of difficult not to talk in a mechanistic way, And so this is the paradigm that we're talking about. But fortunately, scientific inquiry, and I'm a romantic scientist, I believe in, scientific inquiry, it has its own momentum. And by the beginning of the past century, the science of the organism was already emerging in place of the science of mechanism. And there are 3 books that influenced me most of all. They were all inspired by quantum theory. And quantum theory is amazing because it counteracts all your mechanistic intuitions. Now, Alfred North Whitehead, a philosopher and mathematician, science in the modern world, is a very eloquent critique of the flat, colorless, soundless Newtonian universe. And it's all of a piece with Henri Bergson's Time and Free Will. Bergson was inviting us to recover this multidimensional and heterogeneous time that we have that colors all our most intimate experience, making it unique for every occasion. And Schrödinger was struggling against the Copenhagen interpretation of the quantum theory, which says the mathematics and the physics has nothing to say about life whatsoever. But he insisted that, of course, it has. And he insisted that it can even tell you about the meaning of life. And, this book is very, very famous for having predicted DNA as a genetic material, but more significantly, in my view, it predicted the molecular coherence of organisms that was discovered in my laboratory in 1992. Organisms, and you can see these organisms that are from the images on the cover of this wonderful book. I call it my wonderful book because it's my favorite book, and that I have written. Okay, and these are real organisms, life under the polarizing light microscope that geologists use for looking at rock crystals. But real organisms look like dynamic liquid crystal displays. Basically, this is the direct evidence that their molecules are moving very coherently together, so that at every point the light passing through sees static order. And that's why it is the evidence of molecular coherence, quantum jazz, which is the best description of this kind of amazing coherence, which is quantum coherence, which is diverse activities all coordinated without being choreographed, without being controlled. There is no controller versus controlled in this organism. Everything is intercommunicating, and it's basically a molecular democracy of how to make sensationally beautiful music together, okay? I won't have time to talk about this because time is very short. Because if you wanted to hear more about this has tremendous import for holistic health, which would be another long lecture. And Ian said he will invite me back to give this talk. But please look at the Quantum Jazz video, DVD, which is produced in-house. It consists of live images set to music, all produced in-house by a very, very talented Isis bunch. Okay, but what I want to do in the remaining minutes is to say something about the wonderful thermodynamics, which is also very amazing. Well, people say the important thing about the organism is the living system is energy flow. No, it's not. It's energy storage under energy flow. You have to have a means of capturing and storing the energy, and then you have to build a reproductive life cycle that's coupled to the energy flow, and that's what's been achieved. This is the zero entropy ideal of the organism. Now, not only does it minimize waste and distribution, entropy is just waste inside, what it exports to the outside is also minimum. It makes sense because the system depends on the environment for input, and if you foul your environment, then your input is poorer, and you won't be very good. So this is an ideal. If it were true, you will never grow old and you will never die. But the closer you achieve, you get to this ideal, the younger you will become and more healthy. Okay? Right. The reason it can do this is because there are lots of other cycles. Circles are very important inside, and they are all working on reciprocity and cooperation, so that those activities yielding energy are directly coupled, giving energy to those requiring it, and the giving and taking can be reversed. So that's the opposite of the dominant model, the domo economy of maximum entropy that is driven by maximum dissipation and waste. It has no structure, it has no closed cycle. Okay, this applies especially to organic agriculture, agroecology systems. It applies to natural ecosystems as well, but you can have very well managed human systems doing that. And unfortunately, I haven't got time. I will just have to flip over the pictures very quickly to show you the highlights. Basically, what I want to say is that if we go on this circular economy of nature, recycling both energy and resources, we can stop using fossil fuels. We can guarantee food security for all. We can mitigate climate change whatsoever. We launched it in Parliament in the UK. Organic cotton beets, BT cotton in India, we've got the evidence there. Cuba can exist without fossil fuels. Organic urban agriculture is very much the key. Farmers, local farmers in Sahel, have defied all predictions, academic predictions of gloom and doom. They recovered from the amazing drought just by saving tree seedlings and planting trees and creating rain. And Ethiopia, my God, and all this working on circular economy, the innovations of farmers, or else they perfected it over centuries. And you can see all this. And then this George Chen, environmental scientist, went and learned from the peasants in China about the circular economy, and then he perfected the system that I schematized. I call it Dream Farm 1. And look, the pictures from his dream farms, floating gardens, floating rice, so you don't have to plant the seedlings with back-breaking labor, and anaerobic digestion. The pigs are trained to deposit their manure directly into the biogas digester to generate methane for energy and everything, and so their quarters are spotlessly clean, and they're happy, they're smiling, okay? So this this is a more intricate version that we've been proposing for the rest of the world, explicitly combining local energy and food production for mitigating climate change. It can mitigate, just by anaerobic digestion of waste, more than 50% of the energy saving. And then if you incorporate solar, wind, hydroelectric at small local level, you can do without fossil fuels altogether. There is an urban dream farm too being set up in London. Here our journey continues. We are going to launch this report next week, which tells you that we can definitely have green energies. We can have 100% renewables by 2050. We don't need nuclear. We don't need carbon capture and storage. Right. I'll stop here. Sorry. I'm sorry I used up all my time.